Good morning, everyone. We too are doing some work with GSA on more specifically looking at circadian light and personal light exposures and how it might affect health and well-being, more focused on sleep and mood. Our overall hypothesis is that if you are not exposed to enough light during the day or perhaps exposed to too much light in the evening, you might be disrupting yourself. And that circadian disruption may be sort of the underlying mechanism of any sleep or health issues that you might have. So one of the things we learned early on, it's important to measure the personal light exposures because we are also doing some assessment in the building, and what we're seeing is what you measure in the building is just the potential of what you can be exposed to. But it really matters is what you as a person throughout your day is being exposed to. So my talk today will be really the background to what led us to the development of the device that we're using and the metrics that we're using to assess circadian disruption. So it's more of a background to lead to now where we are at that, what Judy mentioned, we are at the translational point where we're using that science to be able to go out in the field and measure something and interpret something with a metric, a health metric that we think it's useful. So that's what the talk will be today. So obviously I'd like to acknowledge Dr. Hairwagon and Dr. Bayer for inviting us to be here, my colleagues at the Lighting Research Center, and some of our project sponsors. Some of the devices and some of the work that we had done had been sponsored by NIH in the past, and we actually were part of the Gene and the Environment initiative. A few years ago was one of the first initiatives looking at this, developing tools for measuring environmental assessment in the field. So we do know that light is the primary synchronizer of circadian rhythms to local position on Earth. So circadian rhythms are every rhythm in our body that repeats at approximately every 24 hours. A great example is the sleep-wake cycle, hormone production. Those are all very important circadian rhythms. And light actually sets the timing of all our biological rhythms and synchronizes us to our local time or our watch. But light, if given at the wrong time or if we don't get enough light during the day, it might also be the major disruptor. So the same amount of light that you're exposed to in the morning is good, but if you expose yourself to it in the evening or in the middle of the night, it might be bad, as what might happen, for example, with shift workers. So the current idea is that circadian disruption is associated with a series of maladies, including poor sleep and higher stress, and we do know that when we don't have a good night's sleep, we're drowsy, we're sleepy. But that's okay. If we get another night of recovery, we're okay with that. The problem is that when we do that over and over and over again, chronically, such as what happens with shift workers, you can start having more serious problems such as increased anxiety and depression, smoking, increased risks for cardiovascular disease, type 2 diabetes, and even higher incidence of cancer. And a lot of that work has been done with shift work, showing that they are, if they work 20 or 30 years in the field, they are at higher risk for certain types of cancer, including breast cancer and colorectal cancer. So what we set out to do, and that's really the background for where we are right now, is can we measure circadian disruption in the field? And if so, how can we do that? So we set out sort of a series of steps where the first step would be to characterize circadian light for humans and for animal studies, and I'll tell you why animals got into the picture, even though we're not necessarily interested in disruption in mice, but mice can be a useful animal model for us to study diseases. The second step would be to develop an instrument to measure circadian light, dark levels, and activity rest pattern levels for humans in the field. The third step would be to quantify circadian entrainment and disruption in the field. So one of the things we learned early on is just measuring circadian light and rest activity patterns didn't necessarily tell us whether someone was disrupted or not. So we had to have a metric that would tease out what disruption was in the field. And then the fourth step would be to build a bridge between actual human conditions to animal models in the lab. A lot of times you can't, well, it's much easier to work with animal models. For example, when you want to do cancer research or diabetes research, animal models are very easy to work with. So the question is, can you translate what humans are being exposed to into animal models and develop patterns of light and dark that would minimize disruption in the field, and then bring that back into the built environment. So it's a way to expedite it. How do you develop those light-dark patterns? Or how do you develop these conditions that humans should be exposed to using animal models? And then the fifth step, it got cut there, was to link that circadian disruption metric, which we're using, we developed in the third step, to health outcomes. Okay, so I'm going to briefly go through all these steps and what we have done to sort of accomplish that. So the first thing was to characterize circadian light for humans. We do know that all we have right now, it's light meters that measure lux. Lux has to do with the response of 2 out of 3 photoreceptors in the eye, and it's associated with one aspect of visibility, which is reading black font on white paper. The spectral sensitivity of, is that a laser pointer? Hey, I pressed something. Never mind. The spectral sensitivity of the visual, or one aspect of the visual system would be the black curve, which is the photopic luminance efficiency function. This is what your regular light meters are calibrated to. The circadian system is a blue sky detector. So measuring lux, you're really not measuring light as it affects the circadian system, because the circadian system has a sort of a short wavelength sensitivity. So we developed a mathematical model that allows us to calculate what we call circadian light and circadian stimulus, which is really the light as it affects the circadian system. And then we developed that transfer function, which predicts the outcome. One of the outcomes would be acute melatonin suppression. Melatonin is a hormone we produce at night in darkness, and light will suppress melatonin. So melatonin is used as a marker of the circadian system. So here we can estimate, if you look at the y-axis here, you have CS, which is circadian stimulus. So circadian stimulus below 0.1, you're basically not activating your circadian system, even though you're probably able to see in the environment, because it's equivalent to about 30 lux at the eye, which is pretty good to see, but you're not activating your circadian system. And then a range between 100 and 300, 500 lux at the eye is where you have in the indoor environment. And then above that is what you have outdoors, which is really saturating. So what you're concerned about is between 100 and 300 lux, which would be a CS of 0.1 to 0.4. And the higher the CS, the better would be to affect the circadian system. So we then developed a calculator where you have the spectral power distribution of the light source, you have the light level, and you can convert the lux into circadian stimulus. So it's a way for designers, architects, or somebody that wants to estimate how much the light source is affecting the circadian system. This is just a tool, and anybody interested, just let me know later on, because this is available on our website. You can use it at your own leisure and give us feedback if you use it. The second step would be to characterize circadian light for animal models, because if we want to do that translation and use animal models, one thing we know right now, light levels we use in animal facilities are 3,000 times higher than we need to, because nocturnal animals are 3,000 to 10,000 times more sensitive to light than humans. So the light that we're giving to animals right now in animal facilities is like exposing ourselves to multiple suns. So we had to then develop this spectral sensitivity function for the animal so that we could translate and give the biologically meaningful light to animals that corresponds to what we humans are being exposed to. And that's what we did here. We developed that spectral sensitivity function. The next step would be to develop this field experiment. And as you can see, we are not very good at ergonomics. We don't really know how to create very comfortable things. We started with very big, bulky things. Where you thought you had head injury when you were wearing those things. Got a little bit better. We tried to get light at the eye because that's what's important for measuring the circadian system. Quickly, we decided that compliance was an issue with trying to put things here at the eye. So we moved on to developing this, what we call swizzle stick. It's actually being used by the NIDA intramural with cocaine users. We just got data back from they wore this for 16 weeks, which was a big accomplishment to get cocaine users to comply with that. But still, it's uncomfortable, and a lot of people don't want to wear it. So we moved on to what we call the dosimeter, and that was actually part of an NIA grant to develop a device to measure circadian light in Alzheimer's disease patients, because we were doing light treatment with Alzheimer's disease. So that's what the dosimeter looks like right now. So we wear it as a pendant, and then at night we put it on the wrist. It does have circadian light, so it's a calibrated light exposure as well as rest activity pattern. So it also measures the rest activity patterns in humans. So it's kind of an actigraphy. Now, what was interesting is that when we did our first study was with the Nurses' Health Study at Harvard Public Health, and we had nurses wearing this, and these are data from the nurses. And one of the things we realized is measuring circadian light in rest activity patterns was probably not enough to distinguish between day shift and rotating shift workers, which we knew rotating shift workers were more disrupted. But when you look at their rest activity patterns or their light-dark patterns, it didn't make that much of a difference because there was a lot of overlap. So the question is, can we develop a metric that would then determine who is disrupted and who is not disrupted in the field? So we came up with that idea of what we call phaser analysis. So if you look at the two graphs, you have the circadian stimulus over the course of 7 days, and then you have the activity rest patterns over the course of 7 days. And basically, you think about two strips of paper, you rotate one with respect to the other, and then you calculate the cross-correlation function between these two outcome measures. One is the input, and one is the output. The input would be the light-dark, and the output would be the rest activity patterns. Then we do some magic with FFTs and all these, get all the smart people to do all the calculations. And what we come up with is what we call a phaser diagram, or in this case would be a phaser magnitude. So a large magnitude means you have a period that runs close to 24 hours, and you are entrained. So you're active when there's light, and you're not active when there's darkness, which is what you want for a diurnal species. Okay. So then we looked at the rotating shift nurses. We do the same analysis. And as you can see, it's already different just by doing that analysis. You can already see the difference between the day shift and the rotating shift. And the rotating shift has a much shorter phaser magnitude. So the way we see it is phaser magnitude may be a good metric. When you collect the data in the field with a dosimeter, you then run the phaser magnitude, and you can determine whether the person is entrained or not just by doing this phaser analysis. So it could be a good way to measure this in the field. So then we looked at the number of nights worked compared to the phaser magnitude. And what we saw is the day shift, even though there was a high variation, it was always higher. And as you increase the number of days worked, you tend to decrease your phaser magnitude, suggesting that you were more disrupted. So there was this relationship that we found that we thought was interesting pursuing. So we've done this, for example, with many other populations. One example is Alzheimer's disease patients. We ran a study where we collected healthy older adults and Alzheimer's disease patients, and these are the phaser analysis for the healthy older adults. So the activity is in red, the circadian stimulus is in blue. This is over the course of 5 days. And you can see that there's a phaser magnitude with the healthy older adults. Alzheimer's patients, which we know are disrupted, have absolutely no phaser magnitude, and they're completely disrupted. So phaser magnitude, again, in another population, seemed to be a good metric for showing circadian disruption in the field. So then the next step is, well, can we translate that into animal models so we can study what's the best light-dark patterns for that? So the first thing we had to do is develop these cage lighting systems because, as I mentioned, the lighting in the labs that we use right now are extremely bright. So we developed this green light, which is the sort of the ideal light for, because they have a maximum sensitivity at that color of light. So that would be the ideal light for the animal to see. And then what we were trying to do, which was the next step, was to use phaser analysis sort of as a Rosetta Stone where you could bridge humans who are diurnal species into animal models which are nocturnal species. So what we did is we looked at the day shift and rotating shift work that worked 1 night and 3 nights, which is what you see on the left. And then we mimic these patterns into the animal. So we developed patterns and we delivered to the animals these patterns that were mimicking day shift, 1 night, and 3 night rotating shift workers. And we measured phaser magnitudes in the animals. And what we saw was that if you look at the phaser magnitude on the bottom here, on the right side, it's the 12-12 would be equivalent to the day shift. The rotating shift schedule, RSS1, would be the 1 night, and the RSS3 would be the 3-night rotating shift. So we see a very similar relationship between animals and humans, even though mice and humans are not the same, that we found a way that we could relate those two and measure these two. And obviously for the phasers, The only difference is that the mice are, because they're nocturnal, they're sort of 180 degrees counterphased. So it seems like we can use the phaser analysis as that bridge between humans and animals. So the last step would be to establish that relationship between phaser analysis, which can be collected in the field and with humans, and one health outcome. And we started with glucose levels, looking at diabetes. So we exposed male mice to light-dark patterns that were simulating day shift, 1-night rotating shift, and 3-night rotating shift schedules. And what we found was extremely interesting. The first thing we found was the way we do it is we collect glucose levels at baseline, then we inject, we administer a bolus of glucose, and then we measure the glucose levels 15, 30, 60, and 120 minutes after that injection of the bolus of glucose. And what we found, as expected, your glucose levels rise after 15 minutes of injection of the bolus of glucose. But what was very interesting is after 30 minutes, the day shift, the glucose levels came down. The rotating shift schedule 1 and rotating shift schedule 2, it actually remained high. What this is suggesting is that people that are working 1 and 3 nights a week, simulated here by the animal, what they were experiencing, they have more glucose intolerance. Okay, so they're not able to clean the glucose from their body as quickly as when you're working 12-hour shifts or day shifts. And then we found this very nice relationship between phaser analysis and the glucose difference. And in this case, we looked at the difference between 30 minutes and 15 minutes. So what this graph here is suggesting is that there might be a very nice relationship between phaser magnitude and your ability to sort of clean up glucose when you eat that chocolate cake in the middle of the night, for example. So here is just a summary of all the steps that we do, and we think that phasers could be a good metric for measuring circadian disruption in the field. So this is just summarizing what we did. What are the next steps? And I think going back to the personal light exposures, I think our next steps is now we want to be able to, each person has their own personal exposure. You have an app on your phone, and then you write a prescription so that that person will maintain entrainment. Now, that phone can talk to your cubicle, change the lighting in your cubicle to give you a personalized light exposure in your home when you get home, or in the entire building. We can decide what we want to do depending on what kind of application we're talking about. But that's what we think the future is going to be. It's going to be a personal device which measures your personal light exposure, and then it connects to your personal environment. So this is sort of the system component. We're actually testing that out, that system in Sweden. We got a project from the Swedish Energy Agency. It's called the Healthy Home, and we're doing that in the homes. So when people come back at home, the lighting in the homes will change to maintain entrainment. But that's not to say that this cannot be done in the office as well, and that's really what we hope to see in the future. So this is the work that, we've been talking to GSA and working with them in these projects, and what GSA would like to do based on the project we have with them is develop non-visual light performance specifications. What do we need in the building to maintain entrainment and alertness? Create and test these recommendations. We had an expert workshop, create best practice guide, test them, and then put it into practice. And I think that's why Judy's so excited about doing the work in these workshops and talking to people about it. So with that, I'd like to thank you for your attention. I think we are going to move out of this fixture lights in the building and move much more into a personal light exposure. So we talked earlier about the computer screen. The computer screen could be a venue for you to deliver light. It could be personal light delivery devices or light removal devices. It could be your own cubicle. I mean, we're doing work with Alzheimer's patients where we're working with a light table. Alzheimer's patients are sitting around the table all day long. So we created a TV that we made it as a light, and that's how we deliver light to them. So I think that now architects, design or specifiers. Now we're talking about much more individualized solution, much more creative solutions. So I think we have to talk to furniture people. We have to talk with displays people and see how we can integrate that into something that can be more personalized.
How about schools?
And schools are very interesting because in general, kids in schools are very regular. So it would be very easy to perhaps do a lighting scheme that would be in the ceiling for schools. But the most important thing about schools, you've got to teach the parents what not to do at night, because you can completely wash out what you did during the day by giving the wrong light at night. So that might be. The other option is you have your own desk. So those are things. So I think that the neat part about it is that I think it's in a time now where lighting can be completely different than what you're used to, is just having a